you saw so much.
Oh, yeah,
Like,
I feel like we're gonna have like that today to do part of the book.
I mean,
No, I can do
I'm
that.
guessing that those
I have a good
worksheets.
feeling that Fisher's gonna be like, you guys, no homework, you gotta worry about your ACT. But really, your homework is
It's
finishing up
gonna
all
be-
the stuff you didn't do.
And it's gonna be due tomorrow, probably.
Yeah, give or take.
But I'm not gonna do it
He's like,
tonight.
what
I have to
is-
mentally
He said he
rest
would-
before the big
Do the
test.
worksheets. Do
For
the,
real though, I do need to do it.
do the paper before the mountain math. The mountain math won't be due
Okay.
until the end of the
This lecture will be short. It should only take me about 20 minutes to get through. I'm just going to talk about how plants move substances in and out of cells. So a little bit of an introduction here. With plants that live on land, they kind of have to overcome some challenges. You know, how do they maintain their water as well as their nutrient balance? Because when they were in the water, they had support. Their water gave them buoyancy and gave them support, but they could easily take in water through osmosis. So how does it provide that structural support for that upright growth? So we'll talk about the vascular system in depth here, how water, minerals, and organic molecules move up and down within a plant, and then how some trees go through secondary growth to obviously achieve great heights. So remember, secondary growth is the presence of a woody stem. So the transport mechanism, obviously plants don't have muscles and they don't have a circulatory system. So there's a couple different ways that they can move substances within itself. First off, water can just go through the cell wall spaces between cells. There are tunnels that connect one cell to another. We call them plasmodesmata. They can move things through their plasma membrane. And then obviously the conducting elements like xylem and phloem that move up and down. But the basic movement of water is that water enters the roots, then it hits the xylem, which only moves like water and dissolved minerals, and then some exit through the stomata as it travels up. The thing about moving materials in the xylem, there's push-in forces as well as pull-in forces. And so the push-in force is basically water pressure entering the roots. And then there's this pull-in force because water molecules stick to each other. That's a property of water. You know, water is cohesive, it's also adhesive. So water molecules like to stick to each other because of their polarity. You have like the negative oxygen and the positive hydrogens interacting with other charges of water molecules. So we have cohesion, water, hanging out together, and then adhesion sticking to the sides or inside walls of the tracheids or even the xylem vessels. So both of these forces interacting to pull water molecules like pearls of a string up through the xylem. And transpiration means just the loss of water vapor through the stomata. So as one water molecule leaves the stomata, it kind of pulls the next one in its previous place. I think I have a diagram that shows this, yes. So here's transpiration, and these are supposed to represent water molecules. So first we have to talk about pushing water into the roots by changing ion concentrations, because water tends to move from high water concentrations to low water concentrations, so it's got to push it into the roots. But then it enters the xylem or the vessel. So we have adhesion and cohesion working together, and then when transpiration occurs, that just means that water vapor is lost through the stomata or the stoma. And as it leaves, like I said, pearls on a string, it pulls the previous bead in its former spot. So lots of interactions going on here. I also like this slide because it shows the water potential gradient. Remember, water has to move from high concentrations to low concentrations, so they filtered in dark blue for the high to low. So water will be moving up here. And so we'll talk a little bit about the mechanisms of changing the water concentration so that water just continues to flow upward. So water can diffuse through the plasma membrane, but it does depend on charged ions and organic compounds. So some form channels to allow molecules to diffuse, moving from high concentrations to low. Some protein channels require energy to move nutrients against concentration gradients. So if you remember, we talked about the sodium-potassium pump like a really long time ago, how ATP is needed to move from concentrations from low to high to climb back up the hill. Well, same kind of thing here, but this time it's ATP dependent on a hydrogen ion pump. So we can pump water from low concentrations to high. So you can actually kind of predict which way water will move just by looking at the solute concentration inside of the organism. So the best way to represent how water moves is looking at its water potential, abbreviated by this pitchfork symbol. I should know the name, but I don't, with a subscript W for water potential. Basically, it's just the potential energy of water molecules. What's your water potential? Is it going to flow up? Is it going to flow down? We tend to look at it as megapascals, or MPa. So regardless of gravity, pressure, concentration of solute particles, water moves from a region where potential is greater, so high concentration, to a region where water potential is lower, high to low. So we have osmosis, movement of just water from high to low, based on concentration of solutes inside the cell as well as outside, looking at it. So when water moves inside a cell, the cell will expand. We have water that presses up against the cell wall. We tend to call this state turgid. So I have a diagram that shows this. So water enters the plant cell. Because of the cell wall, it doesn't cause it to explode. So this is turgid. We have pressure, keeping it very rigid and sturdy. If it was an animal cell and water rushes in and it just keeps rushing from high to low, you could probably burst that animal cell. We have isotonic solutions where equal amounts of water enter and leave the cell. Then we have cases where water leaves the cell. So if we look at just water concentration of solutes, if I have high, bad marker. High H2O, and it's moving out, so that means that there's low H2O. If we look at the solute concentration, that means the solute concentration is low inside the cell, and then outside the cell it's high, because there's higher concentration of solute versus water. So if I'm looking at the turgid, water is moving from high to low, so I have high water out here to low water in here. So that means I have low solutes out here, high solutes inside. So the cell can move solutes inside the cell to create that low water concentration to move water into the cell. So it can control solute concentration. All right, if a plant cell is placed into a solution with very high concentration of sucrose, that's the solute, water leaves the cell and the turgid pressure drops. And so it's kind of what I just talked about on a previous slide, but we tend to call it plasmolysis. I think it was labeled, yep, plasmolyzed right here. So if it loses too much water, the cell can die. If the turgid pressure falls to 0, it tends to wilt. So here's just an example of wilted. Low water concentration inside the cell, it's wilted, it looks really droopy, but if you add water or if it somehow changes its solute concentration so water rushes in, then it just kind of perks right back up. So how do you calculate water potential? Two components to it. First off, you have to look at physical forces. What's the integrity of the cell wall, gravity situation? These physical forces are really, really small. So in fact, they're not even included in calculations. Now, I do want to say that we won't be doing any math with this chapter. I'm sure that if you were, if this was your thing, if you were going into plants, you'd have to calculate that. But for the sake of this class, we're just going to talk about, what are some factors that can go into it. So turgid pressure, the pressure against the cell wall, is considered pressure potential. And if it increases, that means your pressure potential will also increase. So here's a diagram just showing I have distilled water, potential is 0. But if a plant cell, immediately after being put into distilled water, it was 0, but now it's -2 because water is going to be rushing in from high water to low water. Now I have water potential of -2. But then after it's been in distilled water for some time, eventually we will reach equilibrium and it will become turgid, and now our water potential is 0. Okay, the second factor you should look at is the concentration of the solutes. So just looking at what's the solute potential, where is it located, the smallest amount of pressure needed to stop osmosis. Pure water is 0. If you increase your solute concentration, it will decrease your solute potential. So here's a diagram that shows this. With our water potential, P plus S, you can see we have high solute concentration inside the cell, low on the outside. So water will rush in because there's low water here. This is the vice versa, the flip side of it. some more examples. Okay, water moves through osmosis, but it was believed that water just kind of filtered through the lipid bilayer. That's not the case anymore. When this book came out back in like 2007, aquaporins had just been discovered. So now we've known this for over 10 years. But aquaporins are basically pores in the cell membrane that only move water inside and out. Now, we actually read an article about aquaporins with freezing of frogs. Remember what's the term? Freezing of organs. Can't think of the word. It'll come to me. But we discussed aquaporins and how they want to remove water from inside their cells, because if water turns into ice, well, then you pretty much damage the cell. So if you fill it instead with a sugary solution, it kind of lowers their freezing point and they can hibernate. So aquaporins, basically water channels that are found in animals and plants. Speeds of osmosis, but does not change the direction. Water still moves from high to low.
Cryogenically?
Yes, thank you. So these guys received the Nobel Prize in Chemistry back in 2003. Like I said, our book, mentions it briefly, but not much was well known. But basically, they're saying this is an aquaporin, and these are water molecules. And you can see that there's a positive charge inside the channel, which helps attract the water molecules in, and then it kind of repels or pushes it. It's kind of like a magnet flipping on and off. But yeah. Okay, so there is a water potential gradient in the roots and the shoots that enables the transport. So the plant has to control this. If you add too much fertilizer or drought conditions lower, water potential of the soil will limit how much water will flow into that plant. So the plant takes this into account and tries to move solutes around so water continues to flow from the roots up the shoot to the leaves. So evaporation tends to cause a negative pressure or tension and pulls water up from the stem for the roots. There's a transpiration/evaporation, pearls on a string. Transpiration also depends on humidity. If you have high humidity, obviously water vapor won't escape the plant that easily, so then water tends to stay put, and if it doesn't move, well then water is not going to continue to move up. So lots of factors that affect how water moves. All right, so let's look at the root hairs. So I showed you a picture last Friday of root hairs and how their main job is to increase its surface area. And the root hairs always have to be turgid because the water potential is greater than the surrounding soil. They have to tend to use energy to acquire ions, to pump ions to the root hairs to create that low water concentration. So water from outside the soil, which is high, will move into the roots. So a lot of energy expenditure going on in the roots to bring water in from high to low. But there are 3 transport routes that exist inside cells. So I'm going to talk about them and show you a picture. Apoblast, symplast, and transmembrane route. So apoblast means it's going to move through the cell walls and the space between the cells. Basically, we're not going to go through the membrane. So I'll show you a picture of that. Symplast route means it's going to go through the cytoplasm between the cells. Plasmodesmata, that's the key word for symplast, plasmodesmata. And then transmembrane route means it's going to go through the cell membrane as well as the cell walls, maybe the vacuoles within inside the cells. So it's going to kind of go through everything. Now, this is not exclusive. Like if a water molecule or nutrient or whatever it's moving chooses apoblast, it doesn't mean that it's destined to always stay apoblast. It can change pathways at any time. So here is an example of our apoplastic route. We're not going inside the cells. We're going to stay in spaces in between cells. Okay. Our symplast route goes through the plasmodesmata. And then our transmembrane route goes through the cell membrane and the cell wall. Cell membrane, cell wall. So this diagram I think probably does the best job of that. However, when it gets into the endodermis, kind of like the core, it's very selective. So I also mentioned Casparian strips last Friday. Some molecules will reach this Casparian strip and be blocked. And so what it does is it kind of filters it into another direction of where it can move. So if I go back to the slide, it shows the Casparian strip highlighted in red here. Wow. Casparian strip is made of suberin. It's kind of a fatty lipid, kind of repels water, but it's very selective as to how water can move in the endodermis. So some things can come through, some things have to change ways to move in. So it's just kind of a selective barrier. All right, so xylem pressure, like I said, root pressure is really important. We have to use a lot of energy to build up ion concentration so water moves into it, despite transpiration still taking place. But the root pressure at times can be so strong that water will ooze out and literally ooze out of the leaves. We call this guttation or dew. Root pressure alone cannot account for all of the water transport in xylem. Transpiration is the main force for how water moves up in the xylem. Okay, vessels and tracheids also help with the bulk flow. Like I said, water has cohesion forces interacting with it, so they tend to form hydrogen bonds with each other. So there's this cohesion tension theory that means that the strength of the column varies inversely with the diameter of the column. Maybe in biology, Mr. Rourke took out capillary tubes? No, maybe. Like little glass tubes, and they varied in width from fat, to middle to really, really small. And then maybe he had some dyed water and he put it in and you're supposed to see how far the water moved up.
Oh, yeah.
I remember something with the dyed water.
I'll just show you what the capillary tube looks like. Something like this. No? No? Okay.
Wait, what are you talking about?
Capillary tubes. No? Okay. So I have this really, really big capillary tube and this really, really small one. And if I had a beaker of water and I dyed the water so I could see it, water would rise up maybe a little bit in the big one, but as I decreased in size, water would travel up higher because of the adhesion and cohesion forces due to the cohesion tension theory. So tracheids and vessels that have tiny diameters are so cohesive, like the cohesive force is so strong, they literally climb up the tube. So that's why tracheids and vessels are really, really small in plants, because water can go great distances. I mean, it's crazy to think that we have trees that are over 100 meters in height. How does water travel up that trunk without any, pumps, It's amazing. Okay.
With the diameter skinnier, you just needed less water to go up higher? Or are you talking about the dye would climb up?
The water in the, because the dye is part of the water, just to show you. Because you can't, water is clear, so sometimes it's hard to see where exactly it stops. All right, however, there's a downside to this. In order for the tensile strength to continue to work, to continue to move water up through cohesion, it has to be continuous. The water column has to be continuous. And if you get air bubbles introduced into the column, your cohesion will fail. It's like a break in the necklace. And so something called cavitations stop water transport, and they can lead to dehydration because it's just like taking out tracks of a railroad and your train just stops there. So death to part or all of that plant if a tiny microscopic air bubble got in there. So
Come to the water so back up again.
it would have to require a lot of pressure from below to push it up, but the plant doesn't know that. So. All right, the rate of transpiration. So transpiration is water, vapor, water molecules escaping through the stomata. So we know that the stomata opens and closes to help balance water as well as CO2 needs, because that's how CO2 enters. So more than 90% of water taken in roots is lost to the atmosphere. Photosynthesis requires CO2 to enter. So there's some conflicting requirements. How do we minimize our loss of water? Because we're opening our stomata to bring in CO2, and the answer is a stomata and a cuticle to help reduce this loss. So turgor pressure is responsible for opening the stomata. So the stomata is made up of two cells called guard cells, and they open and close based on their turgor pressure. A closed stomata is caused by low water in the vacuole of these guard cells. So the guard cells are flaccid. If you have an increase of potassium ions or chloride ions and maybe some malates, Through active transport, well, now your solute concentration increases, causing a low water pressure, and water will rush inside these guard cells. So water rushes inside, and they become turgid, and then they open up like a giant mouth. So this diagram does a great job here. You can see it's closed or flaccid, okay? We pump in ions to create that low water concentration. Water rushes in, becomes turgid, and it's open. And then when it's done bringing in items, okay, get rid of these ions, and then they go back to limp and they close. So, more diagrams showing the movement of water. So, moving water in, opens the stomata, moving water out, closes the stomata. Okay, so plants can obviously go through stressful times, especially with the amounts of water. So there are some adaptations to get over these droughts. So they can regulate the opening and closing of their stomata for that immediate response. But some plants have some morphological adaptations for longer periods of droughts. So the loss of leaves, they'll just drop their leaves to limit transpiration. If I don't have leaves, that means I don't have any stomata, so I'm not going to lose water through transpiration. Some plants have very thick or hard leaves with very few stomata, and some leaves are covered with trichomes, tiny little hairs that just help reflect sunlight and reduce the heat load of the plants. Some plants can also change their hormonal conditions for even more long-term adaptation. So flooding rapidly depletes the amount of oxygen in soil, interferes with the transport of minerals and carbs in the roots, so you get some abnormal growth results, hormone levels do change. So then what they'll do is maybe they'll respond by forming larger lenticles, basically to help facilitate this gas exchange. We call this parenchyma, loose parenchyma tissue with large air spaces that store and transport gas when the root is submerged. So, okay, the last thing I want to talk about is sugar transport, okay, the phloem. So most carbs, aka glucose, sucrose, made in the leaves are distributed through the phloem to the rest of the plants. We call this translocation. So moving sugar is called translocation. The carb and nutrient-rich fluid is called sap. Anyone here have maple trees? Anyone tap maple trees? Sap was running, I think, a week and a half ago. So I have some relatives that live in the city and they have huge maple trees in their front yard. So one year they started to tap the maple trees and the city of St. Paul got pissed at them. They said they couldn't do that. So
I saw someone tapping a tree in town.
Yeah, there's a lot of town.
okay. Organic molecules are transported up and down the plant, so it's not just, from the leaves down to the roots for storage. They can go up and down. When aphids pierce into the phloem cells, the sap continues to flow. Hormones are also transported in the sap too, so just keep that in mind. Now, in order to move sap, you also have to look at the turgor pressure differences. So we have this pressure flow theory, how carbs in solution move through the phloem. So our carbs are dissolved in that sugary sap, and they have to be released at a sink where they're utilized. So the carb sources are the photosynthetic tissues, which are leaves. Food storage tissues or growing tips are the roots. We call them the sinks. So we have to transport it to a sink. So we call this phloem loading. Carbs enter the sieve tubes, which is basically phloem, in the smallest veins at the source, and most travel through apoplastic, some symplast movements to move. They'll have to use a sucrose and a hydrogen symporter, which is a protein channel. There's companion cells and parenchyma cells that provide ATP to drive this transport from the leaves down to the roots. There's no energy needed for the movement within the sieve tubes. So this is a diagram, I think, that best describes this. Here I have my leaf cell, that's the source of my sugar, my glucose, and I need to bring it down to the sink. Now, the only way to move sugars in a plant is you have to take two glucose molecules and bind them together. So sugar in plants can only be moved as sucrose, which is just 2 glucose. So that goes into a companion cell that helps load it into the phloem, and it travels down the sieve tube to another companion cell to where it needs to go, in this case, a root cell, the sink. So as water is moving down, water moves down the phloem, it drops off its load, and then it recycles back over to the xylem, moving from high to low, where it can help pick up some more sucrose and bring it back down to the roots. So this diagram just talks about the pressure flow that's involved with it. source cell, that's our leaf. How it enters, says step 1, sugars are actively transported from source cells into the seed tube elements. Because of the high concentration of sugar in the phloem, water diffuses into the seed tube elements, raising the water pressure. Pressure causes the sap, sugar water, to flow through the phloem, where they're transported out into sink cells. The water diffuses back into the xylem. Because of water pressure from high to low, it will travel back up and then can diffuse back across to the phloem. So recycle some water molecules. But that's water transport in a nutshell. Oh, and food.